MSc Zoology Fish Ecology. Unit 4. Culture of asexual or sterile fish, hybridization, gynogenesis, androgenesis, transgenic fish, fish conservation and cryopreservation.
Introduction. Fisheries and aquaculture rely on various advanced biological techniques to improve fish production, genetic diversity, and conservation. Some of the key techniques include culture of sterile fish, hybridization, gynogenesis, androgenesis, and transgenics. Additionally, the conservation of threatened freshwater fish species is essential for maintaining biodiversity. Cryopreservation has emerged as an effective method for preserving fish genetic material for future breeding programs. This lecture covers these key aspects of fish biology, highlighting their applications in aquaculture, fisheries management, and conservation biology. Culture of asexual or sterile fish. Sterile fish are genetically modified or naturally occurring fish that cannot reproduce. Culturing sterile fish in aquaculture prevents uncontrolled breeding and reduces ecological risks of invasive species. Methods of producing sterile fish. Triploidy, induced by treating fish eggs with heat shock, cold shock, or chemical agents, e.g. colchicine, to produce fish with an extra chromosome set, making them sterile. Hormonal sterilization. Application of sex hormones, e.g., 17-alpha-methyltestosterone, to induce sterility in cultured fish. Genetic manipulation. Gene editing techniques to produce sterile fish for ecological and aquaculture purposes. Applications. Prevents breeding in aquaculture systems to maintain desired growth rates. Controls populations of invasive fish species. Reduces interbreeding with wild fish populations, preserving genetic integrity. Hybridization in fish. Hybridization is a crossbreeding of two different fish species to enhance desirable traits such as faster growth, disease resistance, and adaptability to various environments. Examples of hybrid fish. Hybrid catfish, Clarias gariepennis times Heterobranchus bidorsalis, known for rapid growth and resilience. Tiger muskie, Esox maskinongi times Esox lucius, a sterile hybrid valued in sport fishing. Hybrids in tilapia, Oreochromis niloticus times Oreochromis aureus, developed for improved aquaculture production. Advantages of hybridization. Enhanced growth rates due to hybrid vigor heterosis. Improved resistance to diseases and environmental stress. Increased production efficiency in commercial aquaculture. Gynogenesis and androgenesis. These are asexual reproductive techniques used in fish breeding to develop offspring with either only maternal gynogenesis or only paternal androgenesis genetic material. Gynogenesis, maternal inheritance, involves activating an egg with sperm DNA that is later destroyed, allowing only maternal genetic material to develop. Induced using UV-irradiated sperm and temperature or pressure shock. Used for producing all-female populations, which are desirable in certain fish species like tilapia and trout due to higher growth rates. Androgenesis, paternal inheritance, involves the removal or inactivation of maternal DNA, allowing the sperm genome to entirely control offspring development. Induced using irradiated eggs where only the paternal DNA contributes to embryonic development. Used in genetic conservation and selective breeding. Applications. Conservation of endangered species through controlled reproduction. Production of genetically uniform fish populations. Development of fish with enhanced commercial traits. Transgenic fish. Transgenic fish are genetically modified organisms, GMOs, in which foreign genes from other species are introduced to enhance desired characteristics. Example of transgenic fish. Glowfish, fluorescent fish created by inserting genes from jellyfish and corals into zebrafish. Growth-enhanced Atlantic salmon, engineered with a growth hormone gene from Chinook salmon to increase growth rates. Advantages. Faster growth for increased aquaculture productivity. Disease resistance to improve survival rates. Cold tolerance in certain fish species for farming in colder climates. Concerns and ethical issues. Potential ecological risks if transgenic fish escape into the wild. Genetic pollution affecting natural fish populations. Regulatory and ethical considerations in fish biotechnology. Fish conservation of threatened freshwater fishes. With habitat destruction, pollution, and overfishing, many freshwater fish species face extinction. Conservation strategies are categorized into in situ, on-site, and ex situ off-site approaches. In situ conservation within natural habitat, protected areas and sanctuaries, establishing freshwater reserves to protect endangered fish species, e.g., national parks, Ramsar sites. Restoration of natural habitats, removing pollutants, reducing dam construction, and improving river connectivity. Community-based conservation, involving local communities in sustainable fishing practices. Ex situ conservation, outside natural habitat. Captive breeding and hatcheries, breeding fish in controlled environments to repopulate wild populations. Aquarium-based conservation, maintaining rare species in research aquariums for future reintroduction. Cryopreservation of gametes, freezing fish eggs and sperm for long-term storage and breeding programs. Techniques of cryopreservation. Cryopreservation is a long-term storage of fish sperm, eggs, or embryos at ultra-low temperatures, -196 degrees Celsius in liquid nitrogen. It is widely used for fish breeding, genetic conservation, and aquaculture. Cryopreservation methods. Slow freezing, gradual cooling to prevent ice crystal formation in cells. Vitrification, ultra-fast freezing to convert biological material into a glass-like state without ice crystal formation. Controlled rate freezing, maintaining precise temperature changes for optimal sperm viability. Applications in fish biology. Conservation of endangered fish species through genetic banking. Assisted reproduction in aquaculture to improve selective breeding programs. Preservation of genetic material for future research and stock enhancement. Conclusion. Advancements in fish biology such as hybridization, genetic engineering, conservation techniques, and cryopreservation have revolutionized aquaculture and fisheries management. These techniques help enhance fish productivity, conserve biodiversity, and maintain sustainable aquatic ecosystems. Key takeaways. Sterile fish and hybrids are useful in aquaculture and fisheries management. Gynogenesis and androgenesis help in genetic conservation and selective breeding. Transgenic fish offer economic benefits but require careful regulation. Conservation strategies in situ and ex situ protect threatened fish species. Cryopreservation is crucial for long-term genetic preservation.